Histones are small, highly basic proteins that are crucial for packaging eukaryotic DNA into chromatin. Their high content of lysine and arginine residues gives them a strong positive charge, enabling tight electrostatic binding to the negatively charged DNA phosphate backbone. There are 5 main types. The core histones are H2A, H2B, H3, and H4. These form the structural unit of the nucleosome. They assemble into an octamer, which is composed of 2 molecules each of H2A, H2B, H3, and H4. The linker histone is H1. It binds to the DNA entering and exiting the nucleosome core particle and to the linker DNA between nucleosomes, facilitating higher-order chromatin compaction. The nucleosome is the fundamental repeating unit of chromatin. It consists of a histone octamer core, around which approximately 147 base pairs of DNA are wrapped 65 times in a left-handed superhelix. This structure is called the nucleosome core particle. The stretch of DNA connecting one nucleosome core particle to the next is called linker DNA. It is typically 20 to 60 base pairs long, varying by cell type and organism. One molecule of histone H1 binds near the entry and exit point of the linker DNA on the nucleosome core particle. H1 seals the DNA wrap and plays a key role in stabilizing the nucleosome and promoting the next level of compaction. Nucleosomes provide the first and essential level of DNA compaction, reducing the linear length of DNA by a factor of about 7. Without nucleosomes, the enormous length of genomic DNA could not fit within the cell nucleus. Nucleosomes serve as building blocks for higher-order chromatin structures. The association of nucleosomes with H1 facilitates their folding into a more compact 30-nanometer chromatin fiber, according to solenoid or zigzag models. Further coiling and looping lead to highly condensed metaphase chromosomes. While compacting DNA, nucleosomes also regulate access to the genetic information. The tight wrapping of DNA around the histone core physically occludes it from factors involved in transcription, replication, and repair. This inherent repression is dynamically regulated through several mechanisms. First, histone modifications such as acetylation, methylation, and phosphorylation of histone tails alter chromatin structure and recruit regulatory proteins. Second, chromatin remodeling complexes use ATP to slide, evict, or restructure nucleosomes. Third, histone variants like H2A, Z, and H3 can be incorporated into nucleosomes to alter their stability and function. This video about histones and nucleosomes is part of our larger molecular biology for biology students course. The course is entirely free and has more than 100 subtopics inside it. We're excited for you to try it, and the link is in the description.